Hi, I'm Krystin Carlson. I study toxicology at the University of Michigan, and I'm in the PhD program there. One of the funny things about studying toxicology is that the more I try to understand how chemicals damage our health, the more I learn amazing ways our bodies cope with harm. One of these ways is called a kinetic trap. I would like to introduce you to heat shock protein 70. But let's start somewhere else. Here. This is a picture of my daughter. It was taken on the day she was born. She was about 4 hours old. We had to drive with our newborn baby 1 hour to return home. So what did we do with our most precious of cargo? We packaged her up as safely as we could in a car seat. Her car seat even had a special infant insert to better protect her tiny head, neck, and hips. Now, just to assure you, we did make this trip safely, and my daughter continues to ride in a car seat. My point here is that biological systems protect some of their most valuable components with a similar packaging strategy called kinetic traps. It's a comfort to have tools to guard against injury or harm. We buckle and cushion our children into car seats, and cells use kinetic traps. Let's enter the subcellular world of life. Cells have precious cargo that they need to protect with care. Cells are made up of many molecules, such as lipids, carbohydrates, and proteins. Proteins, though, are critical molecules. Proteins form the biomachinery inside cells, without which life as we know it would be impossible. Elimination or injury to valuable proteins reduce a cell's capacity to complete reactions, such as detoxification reactions. Injury to your cells means that you, an organism made up of trillions of cells, may not be functioning at your optimum. As a graduate student in toxicology, this is what I am curious about. How do toxic agents, including physical agents like heat and noise, damage cells and the protein machinery inside them? Well, under conditions where damage can occur, cells have a backup plan, kinetic traps. These operate in the same way as a car seat or a seatbelt. For example, your seatbelt does not prevent all damage from occurring during an accident. Instead, it restrains you so that damage that does occur is lessened. It keeps you from being thrown from the car and sustaining severe damage. In cells, kinetic traps prevent proteins from fully denaturing or becoming unrecognizable to the other molecules they interact with. It's hard to imagine how a protein might become damaged. So take, for example, cooking an egg. The white of the egg, which, by the way, is made up primarily of protein, turns from transparent to white as the temperature increases, and the proteins become denatured. The properties of the egg white change along with this denaturation. The wiggly, gooey, clear egg white becomes solid and hard. Here, on the left, is a drawing of a denatured protein. You can imagine that it's unrecognizable compared to the correctly folded protein on the right. Its function will not be as well performed as the folded protein on the right. To prevent denaturation, kinetic traps hold their proteins in a safe state. This allows the protein to return to its correct form once the conditions are favorable. You see, it takes a lot of energy for a cell to produce a functional protein, and if denaturation destroys the protein beyond repair, the cell must start all over and rebuild the protein. However, if the protein can be held in a reversible state with a minimal amount of energy, this benefits the cell. And this is the advantage of the kinetic trap. They operate in single-celled bacteria, fruit flies, and humans. Almost all organisms have kinetic traps that cushion the fragilities of a cell in times of stress. One in particular protein called heat shock protein is a great example. Originally, heat shock proteins were found as proteins created by cells in response to a heat stimulus. That's why they're named heat shock proteins. Heat can cause protein denaturation. Think about cooking an egg. The denaturation of the egg white. Well, the proteins of your cells have regions of hydrophobicity. That means that they don't like water. They're like oil. They're folded up inside of the protein structure. As the protein becomes heated, these hydrophobic regions become exposed. Let's consider a protein like a jelly donut. Upon denaturation, this jelly, which was once kept neatly inside, leaks out. When proteins become denatured, their exposed hydrophobic regions, or the jelly in the donut, are unstable in the water world of the cell. What a mess! Heat shock proteins, however, to continue with this metaphor of jelly donuts, can smell or detect the jelly once it's leaked out, and they bind to it and prevent it from making a mess. Biologically speaking, heat shock proteins bind to exposed hydrophobic regions of denatured proteins. They prevent aggregation and interactions with other hydrophobic molecules. Then, when conditions no longer favor denaturation, heat shock proteins allow proteins to refold and regain their original function. In other words, while protein denaturation creates a mighty tasty egg, for healthy cells, protein denaturation is bad news. Heat shock proteins hold and refold proteins so that damage is reversible. One temperature-sensitive part of the body, or a man's body, is the testis, or testicle. It's of interest to understand how heat shock proteins may protect this area from heat. One study on the testicular cells from rats found that exposure to cadmium, which is a toxic metal, resulted in increases of heat shock protein 70 to increased levels of 250%. This from a toxic metal, not heat this time. Let's take a quick peek at the safety officer of the cell. Here we see heat shock protein 70 in a drawing. You can see that it has this clamping region to clamp down on the ne'er-do-well protein, which you see it doing on the right, which keeps the protein in check. This same protective response can also be seen in animal models. 10 days following exposure to cadmium, rat testes had higher levels of heat shock protein 70, more than 5 times higher than the level found in a control group of rats not exposed to cadmium. Let's move to another part of the body that's shared by both sexes. Exposure to high levels of noise can cause hearing loss. The importance of heat shock protein in protecting your hearing may be surprising to you. In rats exposed to 1 hour of high-level noise compared to rats not exposed, heat shock protein 70 more than doubled. Thus, stress proteins can also be induced by noise. That's noise, heat, and chemicals. Well, this brings me to the coolest study I've ever read. Mice were exposed to heat, bringing their body temperature to 107 degrees Fahrenheit. That's 41.5 degrees Celsius for anyone that's using that scale. That's like a high fever. It was brought to this level for 15 minutes. Then 30 minutes after the sauna treatment, the cochlear, that's inside the ear, expression levels of heat shock protein 70 increased 100 times. Now, the mice receiving this treatment, the heat treatment, along with a control group of mice that were not exposed to heat, were both exposed to damaging levels of noise. A week later, the hearing was tested in these mice, and hearing losses from the two groups were found to differ by 30 decibels. The mice who underwent heat stress before noise gained protection from the noise treatment. They only suffered a 10 decibel hearing loss. This is about quieter than a whisper. While the non-heat treated mice suffered about a 40 decibel hearing loss. That's about the loudness of the rain outside. From these results, it really seems that heat shock protein may offer a protective response. in the hearing system. However, there's a caveat. The mechanism behind this response is not yet fully understood, and other enzymes induced by heat stress could be involved. You see, just because A and B happen together doesn't mean they're always related. Anyways, I'm out of time. Thank you for accompanying me on this journey into the world of cellular living. I hope you will join me next time for another exciting kinetic trap, S-glutathione. That's really difficult to say, so you know it will be fascinating. Remember to buckle up, you and your babies, so those trillions of cells can keep buckling up their proteins. Metaphorically speaking, of course, just to be clear, proteins do not really have seatbelts or car seats. If you have any confusion about this or any questions, you can contact me. My email address is posted below. Thanks and goodbye.